Okay, this is part 3. I just want to quickly say I really should have said this in the other part. Probably should listen to part 2, 3, and even part 4 as close together as you possibly can. So that being said, let's just get started. This is where we left off with part 2. We just talked about how that there are these dark bands that we call A bands, these light bands that we call I bands, and that this whole thing is the muscle cell or muscle fiber, and it is made up of myofibrils. What we're going to do is we're going to take a closer look at that. We're going to take out this one myofibril, blow that up, and we're going to see that a myofibril is made up of myofilaments. So, actually, let me just back up. So one of the things that we want to do is we want to see what is the smallest unit in a skeletal muscle fiber that actually contracts. And I wish I didn't circle that. If you see these lines here, so those lines, if we just take one section here, this is the unit that keeps repeating over and over and over again. And that is called a sarcomere. So it's the smallest contractile unit of a muscle, and it is the region of a myofibril between two successive Z discs. And we're going to take a closer look at that. So it's composed up of myofilaments. As I said, myofibril is made up of myofilaments. These myofilaments have contractile proteins, or are made up of contractile proteins. There are going to be two types, thick filaments and thin filaments. Now, if we go back to the A band and the I band, like I said, this is going to turn into an alphabet soup. The thick filaments extend the entire length of the A band. Thin filaments extend across the I band and partway into the A band. So what we're going to do is we're going to take a look at this on the next slide. So these blue lines are the thin filaments, and the red lines are the thick filaments. So if we see here is our A band, okay, then we see that the thick filaments extend across the entire length of the A band. Thin filaments extend across the I band partway into that A band. Here is this Z disc. So from here to here is a sarcomere. And we'll talk about this M line and this H zone in a little bit. So this is just to kind of, if I take out one myofibril, blow that up, I'm going to back up one slide, then this is what we have, okay? So the Z disc, which anchors, it's between two successive Z discs, are the sarcomeres. The thin filaments anchor to them. And the thin filaments don't overlap the thick filaments in a lighter H zone. And the M lines appear darker, proteins that connect myosin molecules together. So again, I'm going to back up. I'm going to go forward, see if all right, I do have that. So these thin filaments are made up of a protein called actin. These thick filaments are made up of a protein, contractile protein called myosin. So what we see is in this part of the A band, right, because from here to here is the A band, right, because the thick filaments extend the entire length of that A band. So we have this part of the A band here where there are no thin filaments. That is the H zone. What we're going to see is we need to attach these myosin, these proteins, these myosin filaments together. And so we're also going to do that from here to here, and we can't do that over here if there are thin filaments. So the M line appears in the center of the H zone of the A band. Sorry, I did say it was going to be an alphabet soup. So now what we're going to do is we are going to take a closer look at myosin and actin. So these thick filaments are made up of myosin. Each myosin has rod-like tail, two globular heads. I'm not too concerned about that. The big thing here is that the myosin head are called cross bridges, and it has two binding sites, one for actin and one for ATP. That's really the take-home message. So the myosin is going to have a head called a cross bridge, and that cross bridge has a binding site for actin and another one for ATP. So it kind of makes sense then once we take a look at the thin filaments, which are made up of actin, there's going to be a spot for cross bridge formation for that myosin head to attach. So here are these cross bridges or these heads. And let me go forward. Thin filaments, basically made up of actin. Okay. Actin are going to have active sites to which the myosin heads attach during contraction. And we're going to see that they are regulatory subunits that are bound to actin. So let's take a look at this. So, I don't know if you can see. So this is kind of like a whole bunch of beads, right? And those beads are actin. In the center, can you see that there's that dark hole there? That is the active site for that myosin head to attach. So, as long as that active site is exposed, muscle contraction occurs. So then we have to say, okay, what is stopping that myosin head from binding there? And that's tropomyosin. So now if we start to reverse engineer this, you can say, all right, in order for muscle contraction to occur, we have to move tropomyosin out of the way. Once tropomyosin moves out of the way, myosin head can bind, muscle contraction occurs. So what moves tropomyosin out of the way? Troponin. So when something tells troponin, I'm just going to tell you. So calcium binds to troponin. Troponin changes shape. When it changes shape, it moves tropomyosin out of the way. Myosin head can bind to the active site on actin. Muscle contraction occurs. That's an A, not a D. Okay, so I'm going to run through that one more time. So now we know in order for muscle contraction to occur, calcium will be released. It will bind to troponin. Once calcium binds, troponin moves tropomyosin out of the way. Active sites are exposed. Myosin head will bind. Contraction occurs. So now, where does this calcium come from? And that's what we're going to talk about next, in the next part. So finally, hopefully, this H zone, right, because that's where the thin filaments don't overlap, right, this H zone, you can also see this myosin doesn't have any heads, which kind of makes sense. Why would we have a head here when there's no actin to bind to, right? So this myosin head is going to bind. Once that binds, then that's muscle contraction. So like I said, next part, we're going to talk about where the calcium comes from.